Welcome to our presentation on genetic mapping of powdery mildew resistance genes in wheat, guided by experts Stephen Leith and Paul Murphy at North Carolina State University. Wheat is a staple crop worldwide, and its health is crucial for food security. Here, we see various stages of wheat growth, highlighting its importance. Powdery mildew, caused by Plumeria graminis, poses a significant threat to wheat. Utilizing single gene resistance and genetic mapping can help in developing resistant strains. Our primary objectives are to identify molecular markers and map genes responsible for powdery mildew resistance in wheat. The genetic mapping procedure involves phenotype evaluation, primer screening, linkage analysis, and creating a genetic map, each step crucial for understanding resistance. Let's explore the identification and mapping of the PM25 gene, a key player in wheat's resistance to powdery mildew. By crossing specific wheat strains, we obtain segregating populations essential for genetic analysis, paving the way for understanding resistance mechanisms. The detached leaf technique allows us to observe reactions to powdery mildew in controlled conditions, providing insights into resistance levels. We assess the reaction to powdery mildew using a scale from 0 to 9, helping us categorize wheat strains from resistant to susceptible. For genetic analysis, we isolate resistance genes like PM25, crucial for developing mildew-resistant wheat varieties. Primer screening involves bulking segregant analysis, using resistant and susceptible plant groups to identify key genetic markers. Here, we see amplification patterns from RAPD markers, showcasing the genetic differences in wheat materials, crucial for mapping resistance genes. DNA amplification patterns reveal genetic markers in wheat populations, aiding in the identification of resistance genes. We construct a genetic map of the region carrying the PM25 gene, providing a visual representation of genetic resistance pathways. In addition to PM25, we map the PM3B gene, using near isogenic lines and linkage analysis to further understand resistance. Amplification patterns from various wheat lines help identify RAPD markers, essential for mapping resistance genes like PM3B. We analyze infection types in wheat populations, using statistical methods to understand resistance levels and genetic markers. DNA amplification patterns in wheat populations reveal genetic differences, aiding in the identification of resistance genes. Co-segregation analysis helps us understand the relationship between genetic markers and resistance phenotypes, crucial for breeding-resistant wheat. Linkage distance provides insights into the genetic relationship between markers, helping us map resistance genes effectively. Let's begin with a summary of our key findings, setting the stage for a deeper dive into the details. We have identified 16 RAPD markers linked to important genes such as PM1, PM3B, PM12, PM21, PM25, PMTD1, and the PM3 locus. These markers are crucial for understanding genetic linkages. Here, we present a genetic map of the region carrying PM25, constructed from the NK6815*2, NC96BGTA5BC1F1 population. This map highlights the genetic distances and marker identities. This slide illustrates the detailed genetic mapping of specific genes, including PM1, PM3B, and PM12, showcasing the precise locations and distances of various markers. We extend our heartfelt acknowledgments to the esteemed contributors from North Carolina State University, Dr. Stephen Leith, Dr. J. Paul Murphy, Dr. Martin L. Carson, Dr. Ben Wei Liu, and Dr. Rebecca C. Rufty. Thank you for your attention and support throughout this presentation. We appreciate all of you for joining us today.